Pharmapal.
Delivery of Brainshuttle Trontinemab from non-human primates to human efficacy. Hello everyone, and welcome back to our channel. Today, we're diving into the fascinating world of Alzheimer's research, focusing on a recent paper that explores a promising new treatment strategy. We'll break down complex terms and concepts to make them easy to understand. So, grab a cup of coffee, and let's get started. First, let's talk about Alzheimer's disease. This is a progressive neurological disorder that affects memory and cognitive function. One of the key players in this condition is beta amyloid, a protein that forms plaques in the brain. Researchers are still debating whether the accumulation of these plaques is a cause or a consequence of the disease. Now, onto the paper. The authors investigated a new treatment called Trontinemab, which is designed to target these beta amyloid plaques. This study is particularly exciting because it compares Trontinemab with another treatment known as gantenerumab. Key objectives: evaluate the efficacy of Trontinemab in reducing amyloid plaques. Understand the pharmacokinetics, how the drug moves through the body, and pharmacodynamics, the effects of the drug, of Trontinemab. Let's break down the methodology used in this study. 1. Animal models. The researchers used non-human primates, NHPs, to assess the safety and effectiveness of the drug. 2. Drug administration. Both Trontinemab and gantenerumab were administered via intravenous injection. 3. Data collection. Blood and cerebrospinal fluid, CSF, samples were collected to measure drug concentrations and effects. Key terms: pharmacokinetics, PK. This refers to how a drug is absorbed, distributed, metabolized, and excreted in the body. Pharmacodynamics, PD. This focuses on the biological effects of the drug and its mechanism of action. The results were promising. Trontinemab demonstrated a significant reduction in amyloid plaques, comparable to gantenerumab, but with a lower dose. This is crucial because it could mean fewer side effects for patients. Summary of findings. Efficacy: Trontinemab reduced amyloid levels effectively. Safety: both treatments were well tolerated in NHPs, with no major adverse effects reported. The study concludes that Trontinemab could be a game-changer in Alzheimer's treatment. Its unique 2+1 bispecific monoclonal antibody design allows it to bind effectively to amyloid plaques while also targeting the transferrin receptor, TfR1, which helps transport the drug across the blood-brain barrier. 2. Key figures explained. Figure 1, molecular structure and binding affinity. What it shows: this figure illustrates the structural design of Trontinemab, a bispecific antibody. Key points: it binds bivalently to A-beta plaques and monovalently to the transferrin receptor 1, TfR1. The binding affinity is crucial for understanding how effectively the drug can target and clear A-beta from the brain. Figure 2, brain uptake model. What it shows: this figure presents the brain uptake model for both antibodies. Key points: it uses a one-compartment model to illustrate how the antibodies distribute in the brain. The equations provided help explain the relationship between plasma concentration and brain uptake. Figure 3, comparative analysis of Kp values. What it shows: this figure compares the brain distribution coefficient, Kp, for Trontinemab and gantenerumab. Key points: Trontinemab shows significantly higher Kp values across various brain tissues, indicating better brain penetration. This is essential for understanding the potential efficacy of the antibody in treating Alzheimer's. 3. Tables breakdown. Table 1, pharmacokinetic parameters. What it contains: this table lists key pharmacokinetic, PK, parameters for both antibodies. Key points: parameters like Cmax, maximum concentration, and AUC, area under the curve, are crucial for determining the drug's effectiveness and safety. The differences in these values highlight the advantages of Trontinemab over gantenerumab. Table 2, clinical trial design. What it contains: this table outlines the design of the clinical trials conducted. Key points: it details the dosing regimens, participant demographics, and endpoints being measured. Understanding the trial design helps contextualize the results presented in the figures. Future directions. Clinical trials. The next step is to conduct human clinical trials to confirm these findings. broader applications, the technology used in Trontinemab could potentially be applied to other therapeutic antibodies. To wrap up, this study opens exciting new avenues for Alzheimer's treatment. As research continues, we hope to see effective therapies that can improve the lives of those affected by this challenging disease. Here are some challenging questions that can spark insightful conversations about recent research in Alzheimer's disease. Let's explore these thought-provoking questions and their brief answers. 1. Is beta amyloid accumulation a cause or consequence of Alzheimer's disease? This question challenges us to consider the role of beta amyloid in Alzheimer's pathology. According to Wang et al., 2016, while beta amyloid accumulation is a hallmark of Alzheimer's, its exact role remains debated. Some studies suggest it may be a consequence of neurodegeneration rather than a direct cause. 2. How do recent FDA approvals impact treatment options for Alzheimer's disease? The FDA's recent approvals, including lecanemab and aducanumab, represent significant advancements in treatment. These therapies aim to reduce amyloid plaques, as discussed in the 2021 Alzheimer's Association report. However, their long-term efficacy and safety remain under scrutiny. 3. What are the implications of accelerated approval pathways for Alzheimer's treatments? Accelerated approval pathways can expedite access to potentially beneficial treatments. However, this raises concerns about the robustness of clinical evidence. As seen with the approval of aducanumab, ongoing studies are crucial to validate these treatments' effectiveness and safety profiles. 4. Can we trust amyloid PET imaging as a reliable diagnostic tool? Amyloid PET imaging is a powerful tool for visualizing amyloid plaques in the brain. However, its reliability is debated. Some researchers argue that the presence of amyloid does not always correlate with clinical symptoms, as highlighted in studies by Van Dyke et al., 2023. 5. What role does the blood-brain barrier play in drug delivery for Alzheimer's therapies? The blood-brain barrier, BBB, poses a significant challenge for drug delivery in Alzheimer's treatments. Understanding how to transport therapies across the BBB effectively is crucial for enhancing treatment efficacy, as discussed in Partridge's research on drug transport mechanisms. Don't miss out on this fascinating journey into the future of brain-targeted therapies. If you found this video helpful, please give us a thumbs up and subscribe for more updates on health and science topics. And as always, feel free to leave your questions or comments below. Thanks for watching, and until next time, stay curious.
Pharmapal.